Okay.
Is it? Can you see that?
Yeah, you can. Yeah. No, this
Is it?
actually good right now.
Lighting's good right now?
Yeah.
Hey, my name is Haylo. I'm a molecular geneticist here at EnviroDNA, and today I have the absolute pleasure of giving you a peek behind the curtains and walking you through our purpose-built facilities. I'd like to start by acknowledging the traditional owners of the lands in which our facilities are built, the Wurundjeri Woi-wurrung people of the Kulin Nations, and pay my respects to elders past and present. Follow. We actually have 4 rooms here and we work in a unidirectional workflow. So that means we start at one end and we work through to the other end. And that's basically because we want to start in the areas that have the least amount of DNA and then end with the most so that we limit contamination.
So what happens in this lab?
So this is the first lab in our unidirectional workflow. And basically it's just where we store everything to keep it super clean. So all of our chemicals and all of our buffers are stored in here, and we make up all of our reactions in here. And the idea is that no DNA, no sample enters here, so there's limited kind of scope for contamination in here.
So how do we keep it clean?
So we keep the lab clean basically by not bringing any DNA in here, and we wear these very fashionable single-use gowns in here. So once we use them, we obviously throw them out. We also work underneath this hood. It's called a laminar flow, and it basically pushes air away from everything you're working on so that DNA and other bits and bobs can't float into your reactions.
Hello, Shan. Hi.
Another thing we do is obviously we work with our lab gowns on. We have gloves that we wear when we're working and touching anything basically. And we also have bleach that we use to sanitize any areas before and after working. This next lab is called the Rare DNA Lab. If I could just get on my lab coat, that'd be great. And basically this is where we process environmental DNA samples. And those samples tend to have like lower yield of DNA. So we have a specific room to process them so that samples with a higher yield don't contaminate them. Okay, so what this machine basically does is it moves very specific and tiny amounts of liquid from A to B. That's, you know, the long and short of it, but that means that I don't need to do this physically. So this here is our -30. This is where we store all of our DNA once we've extracted it. So we have these racks here and all the DNA is stored on plates like this. Okay. So now we're leaving the first 2 labs in our unidirectional workflow. And this whole corridor is actually a bit of an airlock. It has a positive airflow. So that means as I open this door, air will flow outwards into the next lab. And that's because the next lab has a higher amount of DNA in it, and we don't want the DNA floating back into these labs. Okay. This is the DNA lab, and this is where we process samples that have higher amounts of DNA in them, like scat, soil, or tissue. So in comparison to the Rare DNA lab, where we only process eDNA that has lower amounts of DNA in it.
Okay, what does this do?
All right, so this is a DNA extraction robot. And it extracts DNA for us in a really clean manner. It's entirely enclosed. And so this is where we put all of our samples in once we've processed them. Well, pre-processed them, I guess, for DNA extraction. And yeah.
What's this thing?
This is basically just a baby DNA extraction robot. So we have the parent and the child. And this does DNA extraction as well.
What is this?
This is a fume hood. So it's not like the other 2 hoods we had, the laminar flows. Those protect the samples, whereas this protects the user. So it sucks air into it so that if you're working with chemicals or anything with like a noxious gas, you're protected. Underneath this, okay, here is our -80 freezer. It's for long-term storage of DNA, and people sometimes pay for us to keep their stuff safe. So this is where it goes.
Why are we going to make that safe?
It preserves DNA better. That's all I know. So here's where we wash everything up. Basically, all of our utensils and everything that we use throughout the day, we want to sanitize every time we use them. So they get a big old bath in the bleach bath, and then they get washed with water, and then they dry out for our next use.
Shower?
Oh yeah, this is for if you're in strife. It's an emergency shower. and an emergency eye wash, which hopefully we never have to use. So this is the final lab in our unidirectional workflow, and it's the PCR lab. So PCR stands for polymerase chain reaction. It's the technique we use to amplify DNA, so make a lot of DNA from very little amounts of DNA. So obviously this room has the highest amount of DNA in it.
This thing.
This thing?
Yeah.
So this is a gel electrophoresis dock. So we make rectangular jellies essentially, and they have little indentations in them, and we put DNA in that. And then we apply a voltage and the DNA moves to the positive anode, basically dependent on how big it is. So we use this to visualize our DNA, make sure it looks pretty much what we're expecting it to look like. It's kind of a QC step before we sequence things. This is how we actually visualize those gels. So we apply UV light and there's a dye in it that's UV reactive. So you can see here, this is the DNA. This is a known ladder of like known DNA sizes, so we can compare them to the sizes and make sure that the DNA is coming out basically what we expect it to come out as.
What's a microwave for?
For making the gels. Yeah, so we melt the gel down in that. Also, if you're hungry, No. This is another laminar flow hood, so it pushes air away from whatever you're working on basically. So we work under here to not contaminate our DNA with other DNA essentially.
Oh, what's this thing?
Oh, that? Boy, am I so glad you asked. We want to be as sustainable as possible, and obviously with the lab there's a lot of single-use things that we can't get around, but for those things we can recycle, we have diverse recycling streams for our different wastes. So we've got soft plastic, we have cardboard, and also hard plastic, which I don't have a convenient example for you, but here's what cardboard looks like. And yeah, we just try to recycle as much as we can and limit our footprint wherever we can. So these are PCR thermocyclers. So here are two, and this is just two other versions of essentially the same machine. This is where we do PCR. So they apply very specific temperatures to our reactions to help us amplify DNA. So this is the light cycler. It's basically another version of a PCR machine, except this one picks up fluorescence. So it can measure fluorescence that's being generated in the reaction. So this is where we do our assays, our species-specific assays. And basically when DNA is amplified, our probe that's designed specifically to pick up one species will drop off little fluorophores that cause fluorescence. What that looks like in reality are these little curves, and that's DNA being amplified. So this is basically telling us that what we're looking for is in this reaction. Here and here and here. Yeah. Give me a prompt.
What does this machine do?
So this machine is the Illumina MiSeq i100. It's the newest Illumina sequencing platform in chemistry. It's so pretty. I wish I could make it open its doors up, but that's reserved for paying customers. So yeah, this is the newest Illumina sequencing chemistry. It means that we can do all of our metabarcoding in-house. So that's really quick turnaround times, gives a lot of QC and QA steps into our hands so we can have a closer eye on things. Another cool thing about having it in-house is that it uploads the data straight to our servers so that data scientists can then pick it up and analyze it straight away. So that is the end of our facility tour, basically. So these 4 labs are our unidirectional workflow, and that's how I'll basically move through the facilities every single day. Having discrete labs for different things helps us limit contamination and kind of keep every step of the way as clean as we possibly can. Having all of our robots and all of our automation improves our throughput and reduces our human error. So we can process probably around 1,000 samples every week from DNA extraction all the way through to metabarcoding or single species analysis.
Thanks, Haylo.
Yeah, thanks. So that's basically the end of our facility tour. And gold star for whoever counts how many lab coat changes I do every day.